Hello, people. Hmm. How many people? We got 29 people here. Matt Cole says hello. Let's say hello right back. Looks like everyone's hiding except me. That's okay. Turn the chat off for me. All right, we got 32 people out of close to 50. Let's wait a couple more minutes. Am I recording this? I hope not. Oh, I am. Oh well. Have to edit that out. Ooh, reactions. What's this? Oh, we get claps, we get thumbs up, no thumbs down, eh? Closed caption. Stop, pause, recording. Stop recording. Polls. All right, well, tell you what, it's the losers who didn't come today. So let's get started with our presentation. I'm not sure, again, how long this is going to last because typically I do a lot of BSing during writing on the board and stuff, but we're going to get through it all somehow. Who else has chatted me up? Hi, Dr. Slish. Hello there, Esha. You're a good person, Esha. Okay. All right, so let's get ourselves started here.
Are you on a mic or something?
Am I on a mic? Can you hear me? Can everybody hear me? Can someone tell me whether everyone can?
Yes, we can hear you.
Okay, okay. Thank you, Julia. You're humble and lovable. Where am I? Okay. Okay, so we're going to our PowerPoint now. Pharmacodynamics 2. Wait, is this right? Yes, it is. It's the third chapter, but it's pharmacodynamics 2. So this is going to be really where we'll see how the drugs are actually interacting with cells when they're in the body. So it's all about synapses. All our work's going to be in synapses. So first thing to know is what kinds of synapses there are. Let me just escape for a second. I want to pull my notes up so make sure I'm not forgetting something. Okay, so types of synapses. So these are axons over here. So these axons then can synapse with the dendrite of another neuron. So that's an axodendritic. It can synapse with the soma. So this is axosomatic, axon to soma. Or, and we'll see a lot of this with our drugs, our synapses, axoaxonic. So you've got one axon interacting with another axon. So typically, this is normally what we think about in terms of axons, either interacting with the dendrites or the soma. But this is kind of like modulating. So this neuron is modulating this neuron's activity. And we'll see a lot of mechanisms where that happens, a lot of drugs that influence that. So this is your presynaptic axon bulb. This is what is releasing the neurotransmitter. So if you're in my pharmacology of cardiovascular pharm class, we just went over this in more detail than we'll go through here. But basically, in your axon bulb, you've got these vesicles, and these vesicles are coming from the cell body, from the soma, transported down here to this axon bulb. And these vesicles are going to be attached to some cytoskeleton. This brown line is just some kind of cytoskeleton. And the vesicles have got these little purple things, which are pumps, and little blue things that are enzymes. So the pump is going to put precursors for neurotransmitter into that vesicle, and the enzyme is then going to convert it into neurotransmitter. So this is going to fill those vesicles with neurotransmitter. And also, Later, we'll see neurotransmitter being recycled from the cleft and pumped directly into the vesicle, and that will be another way that we get neurotransmitter in the vesicle. So you've got like rows and rows of these things in a row, and the very front row of vesicles is like attached to some cytoskeletal proteins here that hold it in place, and holds it in place right next to this calcium channel. So these yellow things here are voltage-dependent calcium channels. So you've got these vesicles filled with neurotransmitter locked into place here right next to these calcium channels. So to release neurotransmitter then, if we go back and review the last class a little bit, you get stimulation in the soma, and that goes to the axon hillock, which starts an action potential. The action potential there goes all the way down the axon. When it gets to the axon bulb, that's where we are here. So the action potential comes down the membrane. When it hits this presynaptic membrane here, the voltage change from that depolarization of the action potential opens these voltage-dependent calcium channels. So calcium comes in through these channels, and that activates some proteins here that fuse the vesicle to the membrane, and then it just becomes part of the membrane. And when it does, as it fuses to the membrane, you get exocytosis, and that neurotransmitter is released. So we get neurotransmitter here released into the synapse. And you don't see it in this picture, but there's a membrane on the other side of the dendrite or soma or whatever this axon bulb is interacting with. So you've got receptors there. We'll talk about those today as well. So action potential comes down, voltage-dependent calcium channel opens, calcium comes in, vesicles fuse, release neurotransmitter. So every car has got a gas pedal and a brake. That was our gas pedal. What about the brake? How do we shut this off? So as long as there's neurotransmitter here, you're going to be getting response on the postsynaptic side, on the dendrite of the neuron. So to make that stop, you've got this amine pump here in the membrane. So the amine pump's going to suck up neurotransmitter, bring it out of that synapse and back into the cytoplasm here. Now, we'll talk later about 3 different mechanisms for this to happen. This is just one of them, but basically the concentration of neurotransmitter in the synapse is going to decrease, and that will shut off transmission. And then if that neurotransmitter is pumped in the cytoplasm, it can't be released from the cytoplasm. It's kind of a waste there. So what happens then is it gets pumped by these pumps into the vesicles. Now it's recycled and can be used again. And it's important to think about this neurotransmission here as kind of like a pulse. So remember I talked about in the axon generating action potentials. You don't want to think about it as an on-off thing, like you get one action potential, it comes down, you release neurotransmitter, and that's it. It's more like a constant pulse. So every time an action potential comes, you release neurotransmitter, take it back up. Boom, 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 like that. It's very rapid. And the faster those action potentials come, the longer the neurotransmitter is out before it gets taken up, okay? So that's what the neurotransmission is all about. And then also, you know, if you're fusing these vesicles to the membrane, eventually that membrane is going to get bigger and bigger and bigger, and there's no point to that. And then you're going to run out of vesicles too. So another part of the recycling is you get this endocytosis of membrane here back into the synaptic bulb as vesicles, and these vesicles then just go back into line and they get ready to release neurotransmitter again. Okay, that was fun. That was fun. I think I have a tendency to do a lot of the same things in different classes because it becomes important everywhere. So I know I talk about this a lot. Hopefully it's not boring for you. Any questions? I just opened up my chat window. Any questions for me? Comments? Faithful companion, as usual. Can't go anywhere without him. Right, Gus? Good boy. Good boy. Yes, you are. Okay, no questions. We will move on. So here is an animation. Oh, here we are. What's the difference between the axon bulb and the synapse? Okay, so the synapse is a combination of the presynaptic side, which is the axon bulb, the little gap in the middle, and then the postsynaptic side, which is your dendrite. The synapse technically is just the gap. It's and then you've got the presynaptic membrane and the postsynaptic membrane. Okay, does that make sense, Alexis? Okay. All right, let's do a different share. Bum ba bum bum ba ba bum bum ba bum. Do 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 do. No, sorry, this one. Okay. Oh, not this one. Shoot. I'm still working on being better at my shares. PowerPoint, PowerPoint. Zoom meetings. This is it. Okay. So first animation. All right. So can you guys see this animation? You can see my Google, right? Google-ish. Google me. Okay. So here we are. Calcium coming in. Vesicles fusing, and the vesicles from behind come forward. Here's response at the postsynaptic side. And now we're ready to do it again. Let's watch that again. So calcium channels open, calcium comes in, vesicles fuse, vesicles move forward, neurotransmitter here, response there, and neurotransmitter clears up, response stops, and we're ready to do it again. So if you want, we can sit and watch this for the rest of the hour that we have left in class. I'm getting bored already, though. Okay, what's this one? Let's see what's next. Okay, so let's go back and talk about, I'm just looking at my notes real quick. Oh, one more thing, 3.4. There's that. Okay. So another thing about the presynaptic membrane, and this is really important too, this comes into play with a lot of drugs. You've got receptors on the presynaptic side as well. So typically we think about receptors being these postsynaptic receptors that are going to change the voltage of the postsynaptic membrane. But you also have these presynaptic receptors, and these are responding to the same neurotransmitter. And what they're doing is blocking these voltage-dependent calcium channels. So not totally blocking them, but cutting them back so less calcium comes in per action potential. So when that happens, if there's less calcium coming in, there's less neurotransmitter being released. So it's kind of it is a negative feedback system, whereby if you've got a lot of neurotransmitter out here, then it activates these presynaptic autoreceptors and blocks release of more neurotransmitter. So you're not just constantly dumping neurotransmitter out when there's already neurotransmitter there. So those autoreceptors are really important. We'll talk about them a lot. We'll even talk about these mechanisms here that block that calcium channel, usually involving cyclic AMP, but we'll get to that eventually. Okay. Now, removal of neurotransmitter. How is the neurotransmitter removed from the synapse to stop transmission? I've got some animations on this, so let's go to that. So the most common one is what happens with neurotransmitters like dopamine, norepinephrine, and serotonin. Those are three neurotransmitters we'll talk about a lot in this class. This is the way their transmission is stopped. After the neurotransmitter gets released, it binds to postsynaptic receptors and produces a response. And then eventually it's going to get taken back up by this pump and put back into a vesicle to be released again. Some of it will go to the mitochondrion and get broken down. Some of it, if you notice, diffused out of the synapse. So some will just diffuse out of the synapse. Let's watch that again. So neurotransmitter release, activating postsynaptic side, some neurotransmitters diffusing away, some's going back in through the thing, some's going in, and the mitochondria breaks it down. So bottom line being that a mean pump is taking the neurotransmitter back up. Some of it will be recycled and some of it will be degraded. Okay, Preguntas? Nope. Okay. Moving on. Now, another type of mechanism that's seen with, for us, things that we'll see a lot of are glutamate and glycine. I think GABA too, but I'm not sure. But glutamate and glycine definitely. Hang on a second. Yeah. There should be one there for you, Dad. Sorry, guys. That was my dad. I'm in Pennsylvania taking care of him. He's 89. Anyway, what was I talking about? Okay, glycine and glutamate. So what happens is after it's released, it activates that postsynaptic membrane and gets taken up by these astrocytes, which are glial cells, the helper cells. And the glial cells may break it down, but eventually what they're going to do is ship it back into the presynaptic membrane, and then it gets recycled over here. So you can see here in this animation, I'm breaking it apart, and it's getting transported back into the presynaptic neuron, and here it's being synthesized back into neurotransmitter. Let's watch that again. Okay. Neurotransmitter released, response over here, and now taken up by neurons, by the astrocytes, I mean, and recycled back into there. Okay, so that is your glutamate, your glycine, probably your GABA. And then the last type is only acetylcholine. So acetylcholine we'll see in the brain in two different forms, one activating muscarinic receptors and one activating nicotinic receptors. Two different acetylcholine receptors, they're very different. But in both cases, the transmission is ended by the enzyme acetylcholinesterase, this yellow Pac-Man over here. So here we're getting response, and then eventually acetylcholinesterase will break acetylcholine down into acetate and choline, and both those pieces will be transported back into the presynaptic side, back into the vesicles, and then resynthesized into acetylcholine. So this is a really important enzyme here. It's actually not off to the side. It's all the way through the synapse of an acetylcholine releasing neuron. And it's a really important target for nerve toxins, like sarin gas is, and a lot of other neurotoxins that were developed for what do you call chemical warfare. Yeah. So anyway, if you block the acetylcholinesterase, then acetylcholine just builds up in the synapse. So your muscles all start to just twitch out, and then you stop breathing and you die. So I suggest no one get involved in chemical warfare. All right. So do you know any reason we have nicotinic receptors? That's a good question. Just so the body can get rid of it, introduced to the body? No, it has nothing to do with nicotine itself. That's a really interesting question. Why do we have two different, why do we have nicotinic receptors? It's because what we're going to see here is it's a very fast way for neurotransmission to happen. But why, and why does nicotine activate it? That has to do with what we were talking about in chapter 1 with evolution between plants and animals. So some plants like tobacco, I think tomatoes also to a very low extent, but plants in certain classes make nicotine, and it's to stop animals from eating them, because if you overdose in nicotine, it's really nasty. It really makes you sick. So that's why we have nicotinic receptors, and that's why nicotine activates them. Now, why is it we have two different acetylcholine receptors, nicotinic and muscarinic? That I don't know. There are also some plant poisons or fungal poisons anyway that activate or block the muscarinic receptors. And we'll talk about them in the last section under hallucinogens. But yeah, interesting question. But it doesn't have to do with getting rid of the nicotine. You want to figure the nicotinic receptors were there before nicotine evolved, probably, because they were important for neurotransmission, and then the plants turned some other phytochemical into nicotine, and it was very useful to stop animals from eating it. Okay, let me take a quick look at my notes to see where the heck we are at this point. All right, so that's how we stop the neurotransmission. 3 different ways. Okay, so now what happens on the postsynaptic side? So up to now, we've only talked about the presynaptic side of the story. Let's go back to my PowerPoint. Okay, so blah, 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 blah. Okay, so now we're going to talk about the receptors. So the receptors on the postsynaptic side. There's two main types of receptors, ionotropic and metabotropic. Both of them more or less have got this general schematic structure in that you've got a protein sitting in the membrane, and on the outside of the cell, it's got a binding site for the neurotransmitter. Ligand is like a general term for something that will bind there, because we'll see a lot of drugs will bind there too. So we call this the ligand binding site, or the hormone binding site, or whatever. And it fits, if you see the shape of this and the shape of this, they fit together like a lock and a key. So the ligand is the key, goes into the lock of the receptor. And that has to do with the, if you've had organic chemistry, the functional side groups of the organic chemical, and the functional side groups of the amino acid side chains. So you've got things like acids and amines that will be partially charged and interact with each other, kind of bind to each other non-covalently. You'll have acids that are fully charged, like glutamate may have a negative charge. Get near a lysine, which is positively charged, they'll interact and make a salt bridge, and that's a really pretty tight non-covalent bond. So my point being, this shape is going to perfectly fit into the receptor, and when it does, all these little non-specific bonding things add up together to make a pretty good tight fit in there. But what also happens is when that ligand goes in there, it pulls on the protein, and the protein will change shape a little bit here, and that'll translate throughout the whole protein, and then on the inside of the membrane, you get a change. So this site on the inside of the membrane is the activation site, I call it. So it's like this is going to activate some kind of response in the cell. Usually it's going to activate a G protein, and then the G protein will do something. So binding of the ligand to the receptor changes the conformation of the receptor, and then that turns on this G protein. So that's what I always kind of as a shorthand, I'll say activates the receptor. So the drug binds to the receptor and activates it. Basically, it's changing its conformation, so now that interacts with something on the inside. The third main domain here of the receptor is for regulation of the receptor. So kind of an interesting quote I read somewhere, I can't even remember where, it was so long ago, but somebody said that our cells don't respond to the neurotransmitter, they respond to changes in conformation or changes in concentration of the neurotransmitter. So if you overstimulate a cell with that neurotransmitter, it will pull the receptors away from the membrane because it's now gotten used to that level of neurotransmitter, and it's not responding anymore. You have to go to higher levels of neurotransmitter to get a response. So that's kind of, it's responding. If you have a constant stimulation, it pulls those receptors away. So it's responding to the change in concentration, not the actual presence of the ligand there. That's kind of convoluted. We'll talk a lot more about it at the end of this chapter. But basically, for now, what you need to know is you've got a site on that receptor where you'll get feedback inhibition of the receptor. Kinase will come up and put a phosphate on it, and then it'll block the activity of the receptor. Okay, so we've got these 3 different sites. We've got where the ligand binds on the outside, the activation of some internal process on the inside, and then a regulatory site. It's kind of a broad general schematic look at things. But then we've got, in terms of specifics, we've got 2 main types of receptors that are important for us. One are the sodium channels, ligand-activated sodium channels, and we've got two different ones of those. We've got what I was just talking about, our nicotinic receptor, which responds to acetylcholine, and then we've got our glutamate receptor that responds to glutamate. Actually, eventually, we'll start calling them NMDA receptors because there's two different kinds of glutamate receptors. Mainly, we're talking about the NMDA receptors, but we're not going to worry about that now. We'll talk about that when we get into chapter 11 and hallucinogenics. But anyway, you've got, these are inotropic receptors. Inotropic means that the hormone binds the receptor and the receptor is an ion channel and it opens and ions come through. That's our inotropic receptor. One type of inotropic receptor is the sodium channel, and that's our nicotinic receptors and glutamate receptors. The other type of inotropic is a chloride channel. So when we were talking about membrane potential before, we said when sodium comes in, it depolarizes the membrane, right? Inside the membrane is negative. You add positive charge to it, it gets less negative, and that's depolarization. This is the opposite. Here we've got chloride going in, it's negative, it's adding negative charge. So it's, hyperpolarizing, making it even more polarized. We've got two different receptors here, gamma aminobutyric acid, very important. We'll talk about this a lot in this class. The other is glycine, which we don't talk about much. It's in your cerebellum. I'm sure it's all over the brain. The only time we talk about it is the cerebellum, but anyway, it's also activating this inotropic channel, and this inotropic channel is passing chloride and making it more negative. So these two types of receptors, or let's just say the broad class, inotropic receptors, represent 90% of all the activity in your brain. So most of your brain is just combinations of stimulation and inhibition. We talked about this in the last class when we said like a neuron will have inputs from the positive inputs and negative inputs. The positive inputs are things like this, mostly glutamate, some nicotinic receptors. The negative inputs are mostly this GABA and chloride channel. Let's see how these suckers work. Because frankly, I'm really proud of this animation for no good reason. No, this was my first, I did all these animations years ago using Adobe. Uh-oh. Let me get this so Dad doesn't have to, sorry. Hello? Yeah, what's up, Dad? Oh, Mary, okay. So what the hell was I talking about? Where was I going with this? I'm lost now.
About the animation.
So I made a whole lot of animations, and come December, the end of December, I'm really proud of my animation. Way too proud, frankly. But come to the end of December, all those animations are going to be gone because you won't be able to use them anymore because they're getting away from Flash animation. So this is the first programming I did that was in JavaScript. And I'm like, ooh, I can program in JavaScript now. Really can't though, only little things. But anyway, so this is sodium outside, right? And because of your potassium leak, like we said last time, we've got negative charges inside and positive charges outside. So when we add neurotransmitter, binds the receptor, and now you've got sodium going through, and you can see the membrane potential flip. Now it's positive inside and negative outside. So that's our EPSP, excitatory postsynaptic potential. You've got your neurotransmitter being released from the presynaptic neuron, diffusing across the synapse and activating these receptors and producing a potential change. And when the neurotransmitter goes away, it goes back to rest. So that's our EPSP. Excitatory. Isn't it exciting, people? Okay, here's our IPSP, inhibitory postsynaptic potential. Look how much negative charge is here. And now we're going to add some GABA to this. And more chlorides coming in, now we've got more negatives. So that's an inhibitory response. So again, going back to our neuron, you've got excitatory responses and inhibitory responses. The excitatory are like this. neurotransmitter, and then ions coming in. And our inhibitory are like this. And again, they're brief. They're just like a little bit of negativity, bring it back. A little bit of negative, bring it back. Positivity, negativity. So that is our ionotropic channels. Bum, 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 bum. Okay. God. My dog is snoring. I don't know if you can hear that, but he's snoring the other side of the room. Okay. Now, metabotropic receptors. So metabotropic receptors are a lot more complicated. They're not ion channels themselves. They link to ion channels through some kind of metabolic response, okay? So all of our metabotropic receptors are going to be this G protein coupled receptor type, or another way of saying the 7 transmembrane receptor. So you've got 7 transmembrane regions here, and they fold together to look like this. And right down in the middle of all of those is where the ligand goes. So when the ligand comes down in, it'll bind there, it'll change the conformation, and then this little tail out here sticking into the cytoplasm will move, and it will then become available to activate G proteins. And then out here, this is where the regulatory phosphates go. So when we talk about downregulation, this is where the phosphorylation is going to go. Maybe even on this loop too, but always on the inside. So we'll be talking in this class about, we don't talk about too many neurotransmitters, but we talk about norepinephrine, dopamine, serotonin, acetylcholine muscarinic receptors, a few other ones, opioid receptors, and a few other ones at much lower scale, but these are all going to be 7 transmembrane receptors. So they'll have an awful lot of structural similarity in these transmembrane regions, right? So they're almost identical. But then out here where the ligand binds, they'll be very different. So they'll interact with different ligands. And also the G protein part can be different between different receptors because they'll interact with different G proteins. Most of the structure is the same, which again, if you talk about evolution, you know, we probably way back at prokaryotic stage, these things evolved. And since then, based on the first initial structure, many other ones evolved from it. And then the important structural parts, the part that if you change, it's just not going to work anymore, is conserved over time. And then the parts that are important for differences between different receptors, that part changes a little bit. So these ligand binding domains can be different between these receptors, but the structural parts are pretty similar. And that's neither here nor there. That's not important for this class. That's just a little extra special thing you got today. You're welcome. Okay, what do these things do? Most of these things are going to affect cyclic AMP in some way, whether it's presynaptic or postsynaptic. Mainly we're talking about postsynaptic. Mainly when we talk about these in cyclic AMP, actually, we're talking about axoaxonic stuff. But anyway, this, let's change my share. Not that one. Change my share to this one. So this is how the cyclic AMP system works. You've got an inactive receptor, right, that is not interacting with the G protein. And when the ligand comes along, it changes conformation. That activates the G protein, which then comes along. Oh, stop, damn you. Comes along and activates this. So this is AC is adenyl cyclase. AC will then convert, let's back up. Oops. Okay, active receptor activates the G protein. G protein then activates adenyl cyclase. Adenyl cyclase is going to convert ATP into cyclic AMP. Now, cyclic AMP will activate the enzyme PKA, protein kinase A. Protein kinase A then will phosphorylate different proteins, producing a response in the cell. So whenever you hear the term kinase, kinase is an enzyme that uses ATP to put phosphate on something. And phosphate is like this big, ugly, negatively charged group. So when you stick that on a protein, it changes its conformation a lot because it has to either hide those positive charges or it wants to or negative charges or interact with those negative charges. So when this pKa puts a big, ugly phosphate on there, that causes that target protein to change a lot. Now, what target proteins are we talking about? Depends on the cell. In our case, a lot of times it will be like the calcium channel, that presynaptic calcium channel. If you phosphorylate it, more calcium will go through, so you get more neurotransmitter release. So the cell can modulate how much neurotransmitter it's releasing through the system as well. And I think I'm going too far and too fast, so don't get too confused. Right now, all you really need to know is in general how this mechanism works, and we'll build on it later. And then this guy over here, phosphodiesterase, breaks that cyclic AMP down into good old AMP, just breaks the circle, and now it's just regular AMP. All right, let's watch this again without your obnoxious professor talking over it. Okay. All right, so that's the general scheme. Now the next question is, how does that interact on a postsynaptic side to change ion channels and cause a change in the cell? A lot of different ways this can work. Oh, shit, no. Shoot, no. Let's back up a little bit. So one type of receptor will activate a G protein called GS and activate adenyl cyclase. Another receptor, which we'll see a lot of presynaptically in these axoaxonic synapses, will activate a different G protein, GI. I stands for inhibitory. So you'll see this is going to inhibit the effect of, inhibit the production of cyclic G, cyclic A. Okay, we're back to it, activated GI. Boom. Now, if we stimulate with that agonist like we did before, the conformation of AC has changed and now GS has nothing to interact with. And so we're going to have a decrease of cyclic AMP in the cell. Okay. So we'll see this a lot in axon bulbs where you up or down regulate cyclic AMP, and that will affect how much calcium comes in through the calcium channel and how much neurotransmitter is released. You know, you guys, if you can, it would be nice if you could chat every now and then to let me know where you are, because I'm just going to keep on talking, whether you understand what I'm saying or not. And let me take a quick look here, make sure I didn't. I may do the whole damn chapter today if I'm not careful. I know you would hate that. Then we wouldn't have anything to do on Thursday or Wednesday. Okay. Yeah, we're going to go too far today, but that's all right. All right. So now, oops, I did this again. So stop. So we've got receptors that will increase cyclic AMP in a cell and receptors that will decrease cyclic AMP in the cell. How does that relate to channels? These are metabotropic receptors and they will affect ion channels in the cell. Okay, so this is a potassium channel. You can see all these busy bee potassiums inside. Potassium channel is closed, so they're just buzzing around. Now this shouldn't say GS, it's a different G protein, but a ligand can activate a receptor, and that can activate a G protein, and the G protein directly activates the channel, okay? We'll see a lot of this in presynaptic membranes. I don't know if I stop it. I don't think I do. I think it just keeps going at this point. It's on a loop. So let's see it again. So pretty simple mechanism. Ligand activates receptor, receptor activates G protein. It actually changed shape this time. G protein opens the channel, and then calcium starts going through. Or potassium, I'm sorry, potassium. So here's something I didn't say before that's really important. When potassium goes out of the cell, that's going to make the inside more negative, right? You're losing positive charges, so it becomes more negative. That's an IPSP. Make sure when you're reading your notes, make sure you understand this and you get it solid in your head. EPSP versus IPSP. EPSP is excitatory, that's only sodium coming in. IPSP is inhibitory, and that can be either chloride coming in or potassium going out, okay? So separate those two out and make sure you know them both. And in terms of ionotropic versus metabotropic, sodium channels are only ionotropic. You've got your glutamate and your acetylcholine. And on the other side, for IPSPs, you could have chloride, which is ionotropic, or potassium, which is metabotropic. Look at those potassiums go. Jesus. Zip, zip, zip. Okay. What else can happen? You could also have a channel or a situation where the receptor activates a G protein. G protein activates adenyl cyclase, makes cyclic A, and cyclic A opens the channel. Okay, there are channels like this. In the retina of your eye, you have this. So I don't think we're going to talk about them again in the class, but I throw it out there to let you know it is a possibility. And you could also have a situation where you go all the way to protein kinase A, make some cyclic AMP, activate protein kinase A, and now phosphorylate that channel, and that opens it. So when I originally talked about cyclic A, I was talking about protein kinase A phosphorylating target proteins. This would be an example of a target protein, a channel in the membrane that then opens. Oh. And thank you for sharing. Sharing is caring. Okay. Oh, all right, I know what we're doing next. I want to save that for next time. All right, so here we are, 45 minutes into an hour and 15-minute class. We've got a half an hour to kill. Any questions? Let's stop sharing. Let's see your faces. Bring your faces up for me. Bum, ba-bum, bum, ba-bum, ba-bum. There we are. How many of us are there? 37 people. Good. Okay. Any questions about the quiz from last weekend? I haven't even been looking at your quiz score, so hopefully they're going well. I've used these questions a lot, so they're pretty good. No questions or comments about today's lecture? Make sure you're reading the book too, because I'm going kind of fast. There's a lot more detail in the book than what I'm going through here. Yeah, you know, this whole online thing, I don't know about you, but it's a bit of a struggle for me, because normally when I'm right there in front of you, I can see what you're doing, see if you're paying attention, see if you're understanding, and I can interact a lot more. This is kind of hard because I'm not getting a lot of feedback from you. Okay, so, but I've got some questions here. Could you explain EPSP versus IPSP a little more detail? Okay, so let me back up and put a different PowerPoint up so that we can tie yesterday's lecture in with today's lecture. And in the process, we will answer that question. Okay, so let's share. Boom. Okay, remember this from the last lecture we did. This isn't the best figure. Hang on, people. Stay with me here. Okay. Okay, remember when I showed you this picture last time? These little green things are excitatory and the red ones are inhibitory. And this soma's adding all that up. So these green things are mostly glutamate, activating glutamate receptors and sodium coming in, producing an EPSP, okay? So these are EPSPs. These red things are mostly GABA, opening chloride channels, and chloride going in, causing an IPSP. So an EPSP is excitatory postsynaptic potential. So it's like the neurotransmitter goes out, binds the receptor, opens a channel, and then ions go through. That's your EPSP. If it's sodium, it's EPSP. If it's chloride, it's an IPSP. Okay. Is that okay? Matthew? Matt, you want me to talk some more about that? Maybe you could ask another question. Matt Cole. We good?
Yes.
Yes?
Um,
Sort of.
Sort of. Can you explain the path? Like I understand the overall function, but I don't know um how it affects the synapses.
Mm-hmm.
Yeah.
Okay.
So more in detail about like the actual function once the sodium enters or the chlorine enters and the potassium leaves.
Okay. So this is something that seemed really silly last time, but maybe it'll make more sense if we watch it now. Okay, can you guys see the spatial and temporal summation? Can you see? You can see my Google thing? Google Chrome? You guys are seeing this neuron thing again? Yes, okay. So that is an IPSP. That is an action potential coming down the axon and then releasing GABA, and GABA causes chloride to come in, and that's an IPSP. And it passively spreads over the soma. Another IPSP. Now this is a stimulatory neuron. When the action potential comes down, it releases glutamate. Glutamate opens sodium channels. You get a depolarization, and that spreads over the neuron. Okay, so that's our EPSP and IPSP. What do we got going over here? So EPSP, EPSP, IPSP. Does that make sense, Matt?
Yes, it does now. Okay.
Okay. And like I was saying, each one is very brief, doesn't really affect the axon hillock very much. The neurotransmitter goes out, comes back in. So if you want to get a lot of stimulation, you got to get a bunch of action potentials together. And then you get action potentials over here. Okay. So first exam is next Wednesday. I'm not really sure what we're going to do Monday. We'll have to do some kind of review or something. I wanted to get it Wednesday just to get us on a normal weekly track thing. So it is next Wednesday. Thursday we'll finish this chapter. Probably won't take that long, but we'll finish the chapter. Next Monday, I guess I'll have some kind of maybe I'll have a practice test and we can do that together. Sound good? Yes? Okay, good. Anybody got anything else today, or should we call it a day? How many people want to wrap it up? We're done? Okay. I will see you all here next Wednesday, right? Wednesdays.
Yeah.
Okay. Later, dudes.